today's discussion, we are going to discuss myopathy and effects of physical training on skeletal muscle. Can anyone tell me what is the meaning of the term myopathy?
Disease or problem in skeletal muscles.
Disease or problem in skeletal muscle, और कोई myopathy?
The study about the muscles means if someone has problems in muscle areas or something.
Study of muscle problem. और कुछ?
So inflammation of the skeletal muscle.
Inflammation of the skeletal muscle. और कुछ?
Pain in skeletal muscle which occurs due to muscle fibers, not nerves.
What?
Actually, the pain causes due to the muscle fibers, not the main nerves. In my opinion.
Okay. Let's start with the basic definition of myopathy. The myopathy, it's basically a disease of skeletal muscle where the muscle fibers themselves are abnormal. It means in myopathy what we see, there is an abnormality in skeletal muscle. So due to that abnormality, we can see the abnormal behavior of the skeletal muscles too. So what is the abnormal behavior here? The weakness of the skeletal muscle, right? Weakness of the skeletal muscle. Now here in this discussion, we'll explain what are the different type of abnormality that we can see in skeletal muscle, right? And what is the molecular basis of all those different types of skeletal muscle abnormalities or all those different types of myopathies, right? So these are some basic features of myopathy. It is the primary problem of the muscle, but in myopathies, the functions of nerves or nerve fibers is totally normal. There is no abnormality in nerve. There is an abnormality only into the skeletal muscle or skeletal muscle cell or fibers, right? So that myopathy leads to muscle weakness, wasting. Wasting means the loss of muscle mass and body weight. Along with this, we can see the fatigue. Sometimes we also can see the muscle pain. These are the most common features of myopathy, right? Now, for the skeletal muscle abnormality, there are many numbers of causes that we will explain one by one, right? The molecular mechanism of myopathy. The first one. See, this is a type of myopathy where we can see the structural defect of skeletal muscle, right? So this is called sarcolemmal structural defect. We know about the sarcolemma. It is the muscle cell membrane. So it means in myopathy, we can see the defect in the skeletal muscle cell membrane, right? So now, a common example where we can see the structural defect is Duchenne Muscular Dystrophy, which is very common disorders. So what is the features of that Duchenne Muscular Dystrophy or DMD? It is the most severe and most common muscular dystrophy in children. It means this disease are very common in children, right? And it is a specific type of, it is a genetic disease, right? It is an X-linked recessive disease. As it is X-linked recessive, so the percent prevalence of that particular disease is high in male children in comparison to female children, right? So what is the physiological basis of this DMD? Here you can see it generally occurs due to absence of dystrophin protein. So here, now we need to know what is dystrophin proteins and how this dystrophin proteins is important to maintain structural stability of skeletal muscle. So due to the defect into the dystrophin protein or absence of dystrophin proteins, there is a progressive muscle degeneration, right? So we need to know what is the exact functions of dystrophin protein first. Now, see here, this is a dystrophin protein. This is a dystrophin protein. It is a structural protein. So what is the role of that protein? It generally presents in the inside of the muscle cell, and it helps in the connection. It helps in the connection between muscle cell and the muscle cell membrane, right? So why this dystrophin proteins is important? You can see this is the muscle cell membrane and this is the inside the muscle cell, right? So inside the muscle cell there is a dystrophin protein, and that dystrophin protein allows the connection between muscle cell or muscle fiber with the muscle cell membrane, right? So this is where the importance of the dystrophin protein. Sometimes you can see there is a total absence of that dystrophin protein. That is the defect. See what is the role of that dystrophin protein? It protects the muscle cell membranes from stress during contraction. Yes, whenever muscle contracts, muscle always face some physical stress. So to overcome from that physical stress, dystrophin protein played an important role. But in absence of that dystrophin protein, the muscle cell or muscles are unable to handle that physical stress during muscle cell contraction or skeletal muscle contraction. Not only this, it also involves with the transmission of force generated inside the muscles to the outside. It means during muscle contractions, what we see or what we know, it generates force or force of contractions, and the dystrophin proteins actually involves with the transmission of force generated during contractions of the muscles towards the surface or outside of the skeletal muscle. But if there is an absence of that dystrophin proteins, it will automatically affect the transmission of force from inside to the outside of the skeletal muscle, right? Now see, as I mentioned, this DMD is basically occurs due to the absence of dystrophin. So in absence of this dystrophin protein, the sarcolemma becomes fragile. It means muscle cell membrane become fragile. So muscle fibers ruptures during contractions, and due to the ruptures of that muscle cell, we can see there is a more and more calcium ion influx into the muscle cell cytoplasm, right? So you can see calcium floods inside, and due to the excessive amount of calcium ion inside the cell, activates certain types of enzyme, like there is an activation of enzyme proteases. So in presence of that activated form of protease enzymes, it directly attacks the cell, leads to muscle cell death or degeneration of muscle cell. So these are some important physiological results of DMD. One is loss of membrane integrity. Second is creatine kinase level. There is an elevation of creatine kinase level due to the damage of the cell. So due to the damage of the cells, there is an liberation of more amount of creatine kinase. So we can get more amount of creatine kinase, and at the same time loss of force transmissions. So whatever the force generated during the muscle contractions, that force are not transmitted to the outside of the cell. So these are some important features of these sarcolemma structural defects, or these are some important features of DMD. Now another important type is contractile protein abnormalities, which is also known as myofibrillar myopathies, right? So this is again another important type of myopathies. So in previous case we have seen there is a defect in the sarcolemma, but the features of myofibrillar myopathy is there is a defect into the protein, that is contractile protein. So the most important contractile proteins are actin and myosin. Other than these actin and myosin, we can see some other structural proteins also may be defect into this myofibrillar myopathies. You can see the main contractile proteins affected includes myosin heavy chain, actin, then desmin, nebulin, and tropomyosin. So these are important muscle cell protein or skeletal muscle cell protein. So defect into this protein leads to the developments of myofibrillar myopathy. Now see what is the features of this myosin heavy chain defect. Generally we know the myosin heavy chains, which is a thick filament, right? And that thick filament is very vital for the muscle contraction, right? So contractions of the muscles in presence of normal thick filament, it generates a sufficient amount of force, right? But if there is any mutations into that particular myosin chain or the gene which produces the myosin protein, right? So it affects the overall contractile property of the myosin, leads to the decrease in the force of contraction. So it means the mutation into this protein leads to effect into the contractions of skeletal muscle. There is a reduced force of contraction or generation of force is less. Similarly, we can see the actin is another important contractile protein, which is a thin filament, right? So again, this thin actin filament is also very vital for the actin-myosin interaction, which is required for the muscle contraction. But due to the mutation into this actin protein or the gene which produces the actin proteins, again that affects the cross bridge formation. We know the importance of cross bridge cycling for the muscle contractions, but the defective thin filaments affects the cross bridge cycling. So there is a weak cross bridge cycling. So weak cross bridge cycling means the contractions or force of contractions also will be weak or less, or generation of force also will be less. Now another important type of protein is Desmin. So what is the feature of the Desmin? See, Desmin, which is a cytoskeletal proteins that aligns and encodes sarcomeres. So we have already seen in skeletal muscles there is a specific arrangement of sarcomere. So in a specific sarcomeres, so both actin and myosins are alternatively arranged, and due to this alternative arrangements of actin and myosin proteins, it gives a striated pattern of skeletal muscle. But due to the defects into the skeletal muscle proteins, it affects the alignment of sarcomere. You can see, so the alignment or encodes of sarcomeres basically depends on the protein Desmin. But again, due to the mutations, there is a misaligned sarcomere. So aligned sarcomere is again important to generate a strong force of contraction. But if there is a misalignment into the sarcomere, muscle becomes weak and disorganizations. So muscle becomes weak means again force of contractions also will be less or weak. Nebulin, which regulates the length of actin filaments. We have seen the actin filaments is attached into the jet disc in both the side of the sarcomere, right? It has a specific length, and that length of actin filament is very vital to maintain the cross bridge between actin and myosin, right? The interactions between actin and myosin. But sometimes you can see there is changes in the length of actin filaments. So due to the changes in the actin filament length, muscle becomes or the sarcomere becomes instable, right? And due to the instability of the sarcomere, the force generations or force of contractions is weak. And then again, another important type of proteins is tropomyosin, right? So these tropomyosins generally regulates the actin-myosin interaction in normal conditions, but in presence of some abnormality like mutations causes improper contraction regulations, means the regulations are not well controlled in presence of abnormal tropomyosins or mutated tropomyosin. So these are some important protein which generally involves or mutations of the genes producing these proteins leads to developments of myofibrillar myopathy. Another important type of disorders we can see in skeletal muscle is called excitation-contraction coupling disorder, right? So these disorders occurs when the signals from the nerves reaches the muscle, but the muscles cannot properly release calcium to contract. So what is the most important requirement for the excitation-contraction coupling? The release of sufficient amount of calcium, right? So for the release of calcium, the muscle gets stimuli through nerve fiber, right? So यहाँ पर क्या बोल रहे हैं? The disorders occurs when the signals from the nerves reaches the muscles, but the muscles cannot properly release the calcium. So there is no abnormality into the nerve fiber. Instead, there is an abnormality in the release of calcium ion, right? Now, what are the most important proteins that involves in the excitation-contraction coupling? We have seen about the role of ryanodine receptors. We have seen about the locations of the ryanodine receptor. What is the location of ryanodine receptor? What is the location? Where it is located? Ryanodine receptor, sarcoplasmic reticulum membrane, right? And what is the location of dihydropyridine receptor? What is the location? Dihydropyridine T-tubular system, right? And what is the location of SERCA pump? That is also located into the membrane of SR, right? So all these three protein, receptor means protein, pump means proteins. All these three proteins are vital to maintain the optimum level of calcium ion, which is required for the skeletal muscle contraction. But in excitation-contraction coupling disorders, we can see the defective state of all those three protein. ठीक है, जैसे कि आप देखो ryanodine receptors, which is located into the sarcoplasmic reticulum, right? And what is the role of that protein? Ryanodine receptors, it releases calcium ions into the muscle cell cytoplasms when muscles is stimulated, right? Now, so in normal conditions, if there is a sufficient release of calcium ion from sarcoplasmic reticulum, there will be a muscle contraction due to sufficient availability of calcium. But if there is any mutations on that particular gene, there is an insufficient calcium release. So insufficient calcium release means in the muscle cell cytoplasm, there will be less amount of calcium ion. So the force of contractions also will be less. This is the most common defects in excitation contraction coupling disorders. So तुम लोग को अभी पता होना चाहिए. So what is the disorders? Name of the disorders? This is excitation contraction coupling disorders. The most affected part is a receptors. Receptors is mutated here. What type of receptors? That is ryanodine receptors. What is the locations of that receptors? The location is sarcoplasmic reticulum membrane. Right? Now, in the diseased conditions, these receptors gets defective due to the mutations, right? So that ultimately affects the sufficient release of calcium ion. Now another important thing is SERCA pump. SERCA pumps is vital to maintains the optimum amount of calcium ion. Generally, the SERCA pumps helps in the relaxation of skeletal muscles by removing cytoplasmic calcium ion. साइटोप्लाज्म में जितना सारा कैल्शियम आयन था वो दोबारा सार्कोप्लाज्मिक रेटिकुलम में जाना है. सो फॉर द रिमूवल ऑफ सार्कोप्लाज्मिक calcium ion, cytoplasmic calcium ion, SERCA pump played an important role, and which is very vital for the relaxation of skeletal muscle. Now, mutation leads to the accumulation of calcium ion in cytoplasm. क्योंकि SERCA pump में mutation हो गए. Now that SERCA pumps are not so much active to remove cytoplasmic calcium ion. So इसलिए ज़्यादा से ज़्यादा calcium ion accumulate होने लगते हैं कहाँ cytoplasm में. So which leads to the slow relaxation, muscle stiffness, and muscle becomes fatigue. So these are the two important proteins, and the mutation leads to the defective functions of both the pump as well as the receptors that alters the calcium, cytoplasmic calcium level. Another important one is DHP or dihydropyridine receptors. So what is the locations of that DHP? Again, it is located into the T tubules. So why this dihydropyridine is important? Because it makes a mechanical connection with the ryanodine receptors, and that mechanical connection between dihydropyridine receptor and ryanodine receptors is vital to release the calcium ion from SR to the cytoplasm. But यहाँ पर क्या हो रहा है? This T tubules senses the action potential. It communicates with the ryanodine receptors, but mutation leads to defective communication between these two, right? So बिना communications क्या नहीं होगा? There will be a decreased amount of calcium release. So अभी cytoplasm में the amount of calcium ions will be less. So without calcium ions, the force of muscle contractions also will be weak. I hope this is now clear. Another important type of myopathy is mitochondrial myopathies. So what is the basic definition of mitochondrial myopathies? Mitochondrial myopathies are a group of muscle disorders caused by defects in the mitochondria, which is the powerhouse of the cells, which are responsible for producing ATP. So all of us know about the role of ATP. For the muscle contractions also, ATP played an important role. And that ATP is generated by the active function of mitochondria. But in mitochondrial myopathies, what we can see, there is a defective mitochondria. So this defective state of mitochondria alters the production or generation of ATP, right? See here, these two conditions. This is the normal mitochondria, and that normal mitochondria produce more and more amount of ATP in normal skeletal muscle functions. But in mitochondrial myopathy, means defective mitochondria are unable to produce sufficient amount of ATP. Okay? So without ATP, so the density of the skeletal muscle will be less, right? And that less dense skeletal muscle contractions will not generate so much force during contraction. So यहाँ पर एक चीज मेंशन क्या है? मैटरनल इनहेरिटेंस. सो मैटरनल इनहेरिटेंस के बारे में पता है कुछ? मैटरनल इनहेरिटेंस
स्पर्म का जो हेड मिडिल पीस है जिसमें माइटोकॉन्ड्रिया रहता है वो नहीं जा पाता है. मैटरनल मदर का माइटोकॉन्ड्रिया
inherited from mothers. So as we know that the contribution in an offsprings by a particular parents means there is equal contributions of any characters into the offsprings, right? but there are certain pattern of inheritance which is non-Mendelian inheritance. So mitochondrial inheritance is a type of non-Mendelian inheritance. ठीक है? So यहाँ पर क्या बोलते हैं? In maternal inheritance, mitochondria are inherited only from the mother because the X contributes nearly all cytoplasms. Therefore, mitochondrial DNA disorder shows maternal transmission, not Mendelian transmission. It means in mitochondrial myopathies, if the disorder is present into the infants, so for that the mothers is mainly responsible, right? Because whatever the characteristics that is transmitted to the offsprings, whatever the characteristics related to the mitochondrial functions, that is transmitted from the mothers only. So if the defective mitochondria is there in the infants, and if it is a genetic defect, so for that genetic defects into the offsprings, the mothers is mainly responsible, right? Now you can see here, for the generation of ATP, there are numbers of enzymes required, and those enzymes are important for electron transport chain or for oxidative phosphorylations. Now, the defect in mitochondria means the mitochondrial enzymes unable to act properly in oxidative phosphorylations, they are unable to act properly in electron transport chain. So without proper functions of all those proteins, obviously they will affects the overall generation of ATP, right? So these are some important enzymes or proteins, and due to the defective state of mitochondria, we can see defective functional features of all those proteins like cytochrome oxidase, ATP synthase, and respiratory chain complexes. All those proteins are important proteins or important enzymes for oxidative phosphorylations, for electron transport chain, right? So without them, ATP generation is not possible. So you can see these components normally transfer electrons down the electron transport chain. They pumps the protons to generate proton gradients, and ultimately they involve to produce ATP. But you can see the defects, means defects into these complex, proteins, right? Ultimately leads to the energy crisis in muscles. So without energy, again the contractions will be weak, or generation of force will be weak. So muscles become weak and fatigue very easily. Another important type of myopathy is metabolic myopathies. So we know for the muscle contractions there is an important sources of energy, right? And that sources of energy are utilized by muscle cell during muscle function. Now in metabolic myopathies, we'll see that energy source are not utilized properly by that particular muscle cell. So you can see in metabolic myopathies, it occurs when muscles cannot properly use stored glycogen as a fuel during exercise. Yes, as I mentioned, the sources of energy. यहाँ पर sources of energy क्या है? Stored glycogen. Though there is a availability of the stored glycogen, but the muscle cell are unable to utilize that stored glycogen as a source of energy during exercise. These affects energy productions, especially during anaerobic activity such as running, climbing, as well as weight lifting, right? So इस तरह का physical activities के time बहुत सारे energies चाहिए. So during that time, the body generally depends on anaerobic mode of oxidation, right? So but यहाँ anaerobic activity के ऊपर depend करते हैं. But यहाँ पर though there is a availability of glycogen, but the muscles are unable to utilize that glycogen as a source of fuel during that type of activities or exercise. सो यहाँ पर मेटाबॉलिक मायोपैथीज में sometimes you can see the absence of certain important metabolic enzymes, right? Due to the absence of those metabolic enzymes, the metabolism of the sources of energy are not possible. So ऐसे ही कुछ examples you can see from here. So these are some disorders that are part of metabolic myopathies like myocardial or GSD5 and Palm's disease, right? So in case of first one, you can see the missing enzyme is myophosphorylase, which is a metabolic enzyme, and that metabolic enzyme is vital for the metabolism of glycogen. So you can see without these enzymes, glycogen cannot break down into glucose, and without the presence of glucose, glycolysis cannot possible. So bina glycolysis, the generation as well as utilizations of ATP is not possible. So you can see immediate ATP failure during exercise leads to muscle cramps, and sometimes you can see myoglobinuria, right? So what is the most important features of myocardial or GSD5? The important feature is absence of some metabolic enzyme. So what is the name of that enzyme? Myophosphorylase. So यहाँ पर क्या हो रहा है, देखो मेटाबॉलिक मायोपैथीज में अभी देखा कि ग्लाइकोजन है, बट दैट ग्लाइकोजन्स आर नॉट ब्रेक डाउन इनटू इट्स सिंपलर फॉर्म, राइट? सो क्यों नहीं हो रहा है? इट इज ओनली ड्यू टू द एब्सेंस ऑफ मेटाबॉलिक एंजाइम. ये नहीं हो रहा है, नहीं है. सो इसलिए इसका ब्रेक डाउन नहीं हो रहा है. सो इसलिए ड्यूरिंग एक्सरसाइज मसल इजीली गेट्स फैटिग, क्योंकि यहाँ पर एटीपी सप्लाई नहीं हो रहे थे. सिमिलरली इन केस ऑफ पॉम्स डिजीज or disorders, you can see the missing enzyme is acid maltase, so which is vital for the breakdown of carbohydrate, right? This maltase is important for the breakdown of carbohydrate. Again you can see, so enzymes नहीं है, so इसलिए glycogen accumulate होने लगते हैं. किसमें? glycogen accumulates in the lysosome, and that accumulated glycogens inside lysosome ultimately affects the muscle cell, क्योंकि lysosome में भी lysosomal enzymes होते हैं. At the same time there is an accumulation, excessive accumulations of glycogen. Both these conditions affects the muscle cell, leads to muscle cell damage or distractions. So यहाँ पर भी हम लोग देख सकते हैं ATP falls because muscle fibers are already lost here. So these are two common types of metabolic myopathies. Another important side, other than stored glycogen or other than glucose, we can see the sources of energy is fatty acid, है ना? तो skeletal muscle में कभी-कभी ये भी दिखा जाता है, though there is an availability of fatty acid or free fatty acid, but muscles are unable to utilize that energy source too, right? So यहाँ पर देखो लिपिड मेटाबॉलिज्म मायोपैथीज में क्या हो रहा है? These are disorders where muscles cannot use fatty acids for energy, especially during fasting conditions, prolonged and during illness. So fatty acids normally provides major ATP for the muscles during prolonged exercise, fasting, and events in low glucose state. So even यहाँ पर भी देखो the fatty acids are available, but the muscle cell unable to utilize that fuels as a source of energy. So if fatty acids cannot enter the mitochondria, muscle suffers with the energy crisis, right? So the last part of the myopathies is inflammatory myopathies. So what is the features of the inflammatory myopathies? This is basically an autoimmune conditions, right? So in this autoimmune disease, so the body's immune system attacks its own skeletal muscle. देखो, there are numbers of autoimmune disorders. So यहाँ पर एक देख रहा हूँ हम लोग की skeletal muscles में भी इस तरह का autoimmune disorders देख सकते हो. So in inflammatory myopathies, it is an autoimmune disease. So the body's immune systems directly attacks the skeletal muscle cell, leading to muscle weakness, muscle pain, and even sometimes you can see the overall destruction of the muscle fiber or skeletal muscle cell.